Okay, so I'm going to do a quick overview. ScHARe is a cloud-based population science data platform. It has much of the social determinants of health data in it. And the reason ScHARe was created was to fill three critical gaps. One, to increase the participation of women and underrepresented populations that have health disparities or identify with health disparities to get them into data science. This is really critical because we all know on this call that there are many, many biases that are occurring out of AI development. So this is one way of getting people at the table to help mitigate biases. The other reason that the ScHARe platform was created is to actually create a paradigm shift in the way that we do health disparities, health outcomes, healthcare delivery research, and utilizing new tools and big data that is available to us. This kind of approach allows us to do much more complicated analyses and learning, and potentially understanding the role of social determinants of health and health disparities in the way that we actually can gain scientific knowledge. And then, of course, we're always conscious of bias mitigation and ethical inquiry. ScHARe is composed of four main components. We have data sets. There are over 240, mostly focusing on the social determinants of health and behavioral, and I'll go over these in a few minutes, and some disease and disorders. We are in phase 2, which is building a data repository where people can upload their data into the ScHARe platform. It has computation abilities and tools for cloud computing. It is a Google platform. That's why a Gmail account or something related to Google is required to get into it. We also have GitHub, and the NIMHD webpage has a portal. The ScHARe data system has over 240 data sets, and they're categorized by the social determinants of health of CDC. So economic stability, education access and quality, healthcare access and quality, neighborhood and built environment, social and community context. And we've also added health behaviors and some disease and disorders. These Google Cloud public data sets have no PII in them, so they are easily available and accessible today. The other two, the ScHARe hosted data sets, are also national data sets, but they're not provided by Google, so we host them. Some example of that is the Behavioral Risk Survey, BRFIS and URFIS, which is for youth. And then the last piece, the funded data sets will be for data sharing and for grantees. This is in process and should be ready by the beginning of next year. Of course, on the cloud platform, we have many resources available to you for cloud computing strategies like Python and R, and different ways that you can customize your data sets and create workspaces that are just for you. We also deal with bias mitigation strategies, and we'll be doing other types of work besides Think-a-Thons in dealing with this. The repository that is coming is going to be focused on the common data elements. So with core common data elements, the reason this is so important is because these core common data elements allow data sets to be aggregated. And if all of the data sets that are using these core common data elements, then we have a pick and we can choose different types of data sets that we can create big data sets with. Much easier than if you do it post putting in the repository, because then it takes a lot of data cleaning, data munging, data wrangling in order to make these happen. One of the other nice things that we're attempting to do with this is in the core common data elements, we're also mapping them to big project federated data sets like the American Community Survey, BRFIS, so that you can actually take the federated data sets and link them into your project data. I told you I would talk a little bit about Terra. Terra is the interface into the ScHARe platform. It is also the interface of the major cloud platforms at NIH. So many of you might have heard of Anvil, Biodata Catalyst, and all of us. These all three and ScHARe all use the same Terra interface, which then will make it very easy for us to collaborate across platforms as well. So ideally, you can go into Anvil and go into ScHARe and pull data and do epigenetic studies and epigenetic research. In order to utilize and stay up to date with what we do in ScHARe, you can click on the Stay Updated link on the web page, and this is how you get to the web page. Insert your email, and we send out updates and emails about upcoming events through this link. In conjunction with the ScHARe platform, we also do Think-a-Thons, and that's what this is today. So these Think-a-Thons are monthly sessions that are 2.5 hours, 3 hours long. Some are instructional. We're moving into some that are going to be doing research, hands-on research. And these teams will stay together in order to create collaborative research teams, networking, mentoring, and coaching. So it is our way of trying to advance the use of the ScHARe platform, as well as to advance health disparities research. You already took this interest poll, so I want to introduce our guest experts today. Anya, Summer, and Courtney are from NIDDK Central Repository. Rebecca Rodriguez, who's on the call as well, is a leader at NIDDK who's leading a challenge on AI-ready data sets. Today, we're very fortunate that they're going to give us a presentation on how to do AI-ready data. Anna is a public health data analyst from Booz Allen, and she has lots of experience in both health standards, technology, biomedical, semantic standards, metadata. For those of you who were on our last call or our last Think-a-Thon, you know how important the semantic standards are, because with the common data elements, you have to have common definitions in order to link data sets. She completed her bachelor's in biomedical engineering at the University of Virginia. Summer is a PhD computational neuroscientist, and she investigates the boundaries of AI and drives data science solutions for federal government clients. She holds a PhD in complex systems and brain science, and she serves as an artificial intelligence subject matter expert for the Indo-Pacific Defense and Health Projects. Courtney is also a PhD, a health data scientist at Booz Allen. She is supported by the Office of Data Science and Strategy to develop online data science learning resources. So this is very much a part of her role. And she completed her PhD in epidemiology with a focus on causal inference at the University of California, Davis. I want to especially thank Rebecca Rodriguez for getting all of this together in a collaboration with NIMHD as we move forward into this NIDDK CR data-centric challenge. The purpose of the challenge is to enhance the utility of NIDDK data sets that focus on diabetes to generate AI-ready data sets. The reason this is important is because for health disparities, diabetes is a #1 problem for many racial ethnic groups. I'm going to turn it over to our guest speakers now so they can talk about the challenge and move into their presentation. Anya?
Yep. Thank you, Debbie. Can everyone hear me okay?
I'm going to stop share.
Okay.
Yes, Anya, we can hear you.
Okay, great. Thanks. And I believe Summer is going to be sharing her screen. Great. So before we dive into the actual presentation portion on AI readiness, if you go to the next slide, Summer, we wanted to quickly introduce the NIDDK Central Repository. Courtney, myself, and Summer are part of the support team, and we had this opportunity to present at this webinar today for all of you. And so for those of you who are not aware of the NIDDK Central Repository, it was established in 2003 as a mechanism to provide the broader research community access to clinical research data, as well as biospecimens that were generated from NIDDK-funded multi-center studies. And so this sharing of data and biospecimens allows researchers that were not involved in the original research to test new hypotheses without collection of new data and biospecimens. And so the Central Repository, it adheres to FAIR and trust principles, which you will hear throughout this presentation. And the repository was also recently certified by Core Trust Seal as a trustworthy digital repository. And we encourage all attendees here today to explore the NIDDK Central Repository and the data and biospecimens that we have available. And as Debbie mentioned, a lot of the data that we have is on, focused on diabetes, which is closely tied to social determinants of health. So very interesting data that we have in the repository. Next slide, please. So you all might have seen from the communications from Luca about the data-centric challenge, and this is a challenge that the NIDDK Central Repository is hosting, and the aim of this challenge is to enhance the data that is currently shared through the repository for AI-driven secondary research. And so the Central Repository will conduct a series of these data-centric challenges that are intended to build on one another to develop tools, approaches, models, and methods to increase data interoperability and usability for AI and machine learning applications. And actually, Summer, it's the previous slide from this first. Yep. And so I'm on the third bullet now. For this first data challenge, NIDDK is seeking innovative approaches to enhance the utility of select NIDDK data sets that are currently shared through the repository that are focused on type 1 diabetes. And these studies include the TEDDY study, which is a prospective cohort study that followed children with and without a family history of type 1 diabetes. And this study also explored a lot of social determinants of health factors that may influence disease development. And the other study is TrialNet, which is a network of clinical trials for type 1 diabetes. And so the goal of this challenge is to generate an AI-ready data set that can be used for future data challenges. As I mentioned, these challenges will build on each other in the future, and we're looking to generate more and more AI-ready data sets through these challenges. And then the second goal is to produce methods that can be used to enhance the AI readiness of NIDDK data. And to note, we will be hosting regular office hours during this challenge to provide participants with the opportunity to answer their questions and continue to gain experience in the AI research space. And so those who are interested in participating, they can register for the challenge through challenge.gov, and we've provided the link, and the deadline to register is 5 p.m. November 30th. Next slide. And just quickly to explain the goal and the prompt of the challenge, participation in this challenge will be tiered based on the challenge applicant's experience with data science and analytics. regardless of the participation level, all participants will be instructed to prepare a single merged data set by aggregating all the data files either associated with TEDI or TrialNet, depending on their tier of participation, and then augment that single data set to create an AI-ready data set. And so the beginner challenge will utilize the TEDI data set, and so participants will aggregate and harmonize data sets across this study to create a single AI-ready data set. And then for the more intermediate advanced challenge, participants will aggregate, harmonize, and fuse also data across 4 different studies that are within the same TrialNet network of studies. And this data will be made accessible to participants through the NIDDK Central Repository Analytics Workbench, which provides tools for participants to analyze and access the data. And so this challenge is a great opportunity for all of you to apply the methods and techniques you'll learn today. And so with that, I will switch over to Summer, who will go through the first part of the presentation.
I just want to interrupt 1 second. If you're still having trouble getting into ScHARe, please raise your hand and they're going to put you into a breakout room.
Debbie, we're going to do that in 5 minutes, if that's fine.
Let's try to start now with it.
Okay.
Okay.
Okay. And I think Summer and I'm seeing a prompt to join the breakout room. I think Summer and Courtney might have accidentally joined.
They should be back now, I believe.
Yeah, I was going to say, just people having trouble.
Sorry, I didn't see the slides for a second, so it was throwing me off. So that was the data challenge. And then next slide, please, Summer. I'll just go over quickly the topics for today, and then I'll hand it off to the first presenter. So the goal of this webinar session is to prepare all of you to conduct future AI-assisted research. And so we'll start off by providing an overview of what AI-assisted research is. This includes what constitutes an AI-ready data set and how AI-assisted research is an iterative process. We'll also cover some common AI concepts and their applications. And we'll talk about the importance of a strong research design and subject matter expertise to ensure transparency in research. And we'll also cover the biases that exist in AI, especially in the context of minority health. And finally, the importance of fair and care principles in research. And then we will walk you through, for the second part, walk you through the process of preparing data for AI research using gold standard data management principles, followed by a live demo in the ScHARe platform. And then if there's time at the end, we will cover how to conduct AI research using health data, including how to handle imbalanced data sets, how to select a machine learning model, and then we'll walk through a real example of how AI is being used in the health research space. So with that, I'm going to pass it now to Summer Rankin, who will cover an overview of AI-assisted research. Thank you.
Hey, everyone. I'm Summer, and I'm going to do a quick overview here of AI in health, and then we will get to some more hands-on things with Courtney. So in the interest of time, I may hit some of these slides a bit quickly, but don't worry too much because you're going to be able to do she's going to walk you through a lot of the steps. So we are talking about getting an AI-ready data set. And we first, let's just quickly kind of define what is AI. People use it a lot nowadays. It means, essentially, we like to think of it pretty broadly. We just think of it as, using machines and specifically algorithms to do jobs that would normally require a human. There are different types of AI, different subsets of it, but sort of a general definition is that's how we think about it, right? So we're using the machine to automate things, curate things, recommend, now we have large language models. We can create code. We can do a lot of complex things like, generation of text, but it's not at the point where we're wanting it to take the place of critical thinking, decision-making, and things of that nature, right? So that's kind of the big distinction that I like to make, is that, it allows humans to do what humans are good at, right? Which is the critical thinking piece, using our expertise, using our knowledge. Okay, so I think we have another poll or a fill in the blank. Kevin?
Yep, absolutely. You should see the Slido prompt at this point on the screen.
Yeah,
and we'll
so
start to see some responses trickle in. Yep.
Great. So yeah, the question here is, what do you think are the important features of an AI ready data set? Before I'm, of course, going to give you a definition.
Also, for those of you who can see the screen, if you see there's this little thumbs up thing over there in the corner of the like the topic, it's like if you click on that, it just raises it up. Like many people think that's important. So you don't have to type anything in. You can just vote for someone else's and it just moves it up to
Oh, nice. Good answers. Yeah.
to the answers. So feel free to click on any one of those little thumbs up.
This is great. I love the documented data. Yes. We're going to talk about that. Standardization. Absolutely. Metadata. Standardization. Again, important. Verification. Love it. Yeah, you guys are definitely hitting all of it. Aha, mention of bias free. Reduced bias, yes. Accessible. Robots, okay. Great. Yeah, okay, well.
Summer, I think you can go on.
Yeah, let's move on. Basically, I don't have much to add, really. Let me share my screen again. Okay, so we say, AI readiness is data that's machine readable, reliable, accurate, explainable, predictive, and accessible. And I think you guys actually covered the hive mind covered all of these, which is great to see. So we like to think of data that's reflective of the population from which it's drawn. This is really important. We're going to talk in the next slide about some more detail on that. Data that is well documented and fair. So this is, findable, accessible, interoperable, reusable, model agnostic, right? So it's not, just done for one specific type of model, but it's clean, it's ready to go and ready to be used for really any kind of model. And so we've got pre-processing, handling of missing values, labeling and or recoding, and documentation. And this is all really important, and you're going to do some today. This slide is to sort of talk about the fact that AI research is just like regular scientific research. It's not something that we do in a straight line. And it's not something where we just have an idea and then we stick with that. And then that's like, what you say at the beginning is going to be what you end up doing, right? We all know that you first come up with a question, you then go and investigate what the data is for that question, and you find out maybe the data isn't what you thought, or you couldn't get the data you wanted, or any number of things, right? So you kind of continue to go back and refine and revise those questions. And then even once you go and you do start to get your question down, get your data down, and then you start doing the preparation and the modeling. And the modeling too is itself iterative as well, because you might find that the model you thought was going to be good is not in fact so great. And so that's okay. You know, that's why it's good to have that AI ready data, because you can go and try a lot of different models and evaluation techniques. You talk to some other people, and it's just it's a cycle. It's the kind of the take home message here. And then of course we want to release the data. We want it to be available, and, you know, hopefully we would be publishing or at least releasing the model results as well. So I want to talk about, there's really kind of two or three like major types of machine learning, right? So when we talk about AI, we're really, you know, we can then go to a subset of AI that is called machine learning. And when it comes to health research, that's really what we're referring to for the most part. And so within machine learning, we kind of have two major types, right? We have supervised learning and unsupervised learning. Supervised learning is when we have labels. That's really it. So what I mean by that is that you have data that has a label. So let's say it's your, you know, white blood cell count, right? And maybe that's something that you're trying to predict. If you have, you know, numbers like this, then you're going to, you know, those are your labels, right? That's the thing you're trying to predict with the model. You're then going to use all the other information about, you know, each patient to try and predict the white blood cell count. And so we call all of this other information features. And then the thing you're trying to predict is the target. And we have a couple different ways we can do some supervised learning. We have regression, right? And so regression is where, like the white blood cell count, we have sort of a continuous variable, and we're trying to predict that. Now, if we have something like, let's say I have, you know, a healthcare application, I can take a picture of a mole and it's going to predict whether it's cancer or not, right? We call this a binary classification. So it's a yes, no. That is on the lower left here. You'll see that as, you know, just kind of a different type of supervised learning, because we still have labels, but instead of predicting this, you know, continuous range of values, we're just looking at a yes, no. And so I have a silly example here, which is cats and dogs, but you know, you can see how this could apply to many places in health research. I like to think of deep learning as a subset of supervised learning. There are some nuances there where that's not necessarily always the case, but for the purposes of an overview, that's how we'll think of it. The difference really between, let's say, deep learning and maybe a logistic regression is that we're using an architecture. The algorithmic architecture is based on the way that neurons in the brain work. Loosely, it's loosely based on this. And so what you see here is like, everybody loves to like throw these up and be like, look at my graph. But essentially this is to give you an intuition of kind of what's going on behind the scenes. So instead of having a model that is going to take in all my features and weight them, you know, how it needs to, and then put out the prediction, this is going to have many layers of weights, and we call these hidden layers. And the more hidden layers you have, the more deep your network is. And so that's why this gets referred to as deep learning. And the issue with deep learning, we're going to come back to this later, is that it is not interpretable. Sometimes this is okay. Sometimes it's not. And what I mean by this is that you cannot get the weights out of these hidden layers. You cannot understand in a clear way how the model made its decisions. There are things you can do to kind of interrogate the model after the fact. But when it comes to healthcare data, this is a very important point because it's an important part of mitigating bias. We need to know how the model is working. And so this is why a lot of times you don't see a ton of deep learning. I'm not saying it's never there, but you don't see a ton of deep learning for this reason, because people, especially clinicians, they don't want a model they can't investigate, and trust is really important when it comes to choosing the right model. Okay. Unsupervised learning. Couple of different things we do here. Let's say you have data that does not have any labels. So we had a data set one time, I was working with FDA, and we wanted to detect whether or not somebody had an adverse event after a blood transfusion. So we had all of this data for people that had had a blood transfusion, but we didn't know who had had an adverse event. And it's something that we wanted to pick up things that maybe the doctors and the clinicians had not picked up on, because there was anyway. So the idea of that is that you're looking for an anomaly, right? You're looking for something that's strange. What we actually did there was we took the notes, because we didn't think it would necessarily be in the structured data, which is like all the lab values, the vitals, the weight. We took the text from the notes and we put it into essentially a couple of different clustering algorithms to say, is there a big cluster, or is there kind of this little weird cluster of strange people, right? And then we had epidemiologists investigate those anomalies. So long story long, this is a common thing to do when you don't have labels and you'd like to see, you basically are saying, hey, algorithm, tell me what the groups are in my data, find the patterns for me. And this can be a very useful tool and can sort of teach us things that maybe patterns we wouldn't necessarily see. We did pick up actually some anomalies that were not necessarily noted in the documentation. So it works. Sometimes it works, sometimes it doesn't. Yeah, so that's it for unsupervised. So a couple of examples here, AI and healthcare. These are some of the more kind of well-known ones. A lot has been done around computer vision. And when we use computer vision on things like images. So here I'm showing an example of, I believe these are MRIs, and they were looking at, cancerous versus non-cancerous and various like polycystic, kidney disease. And they were able to give essentially labeled examples to a deep learning computer vision network and say, is this polycystic? Is it not? And once it got a bunch of examples, it was able to tell the difference, right? There are some pretty famous examples of these failing as well, because sometimes it's not picking up on the cancer, it's picking up on like the type, the doctor's name at the bottom, and like because some doctors were submitting more like cancerous patients than others. So you have to be careful with these deep learning models, right? Because you don't know what it's picking up on, you just know it's picking up on something. But some of them are very effective, and it's very interesting research, to say the least. Another kind of big area of like AI is omics, right? Omics is the sequences, and there's a lot of proteomics and genes and all kinds of different sequence learning, and then a lot of these deep. So I've listed a bunch here. These neural networks that are really good at understanding how things are unfolding in time. This isn't necessarily time, but it is still a sequence, right? So these architectures can be really useful. And in fact, they're kind of the same ones we use for ChatGPT. Not necessarily exactly, but because language is also a sequence, these linguistic models are really useful in this omics research. Okay, so even if you're simply preparing an AI-ready data set and maybe you're not ready to model, it's still important to think about research design. Strong research design is really important, right? Because you need to understand the plan, and you need to understand the data and the domain well enough to know what data to collect and whether that data is going to pass muster. So for example, if you are looking to tell, let's go back to the the mole study, right? If we're looking at identifying from pictures, moles that are benign versus cancerous, then we need to understand, okay, well, for what population? Is this for a white population? Okay, fine, then we can do it for, white individuals. Is it for a more diverse population? Well, then the data that we train the model on needs to represent that. And this is a huge problem in research already, in AI already. And now when we put those two things together, you can imagine that this continues to be an issue, which is why we're all here, right? So, ensuring that you have a data source and the details of that data are both collected and represent the population. So for example, let's say you have collected the data, but maybe you didn't even collect what race people were. Is that something you can get? Is that something that you cannot get? Maybe that data set's not appropriate, right? Or maybe there is a data set that has it, right? So even if you're only thinking as far as like the data, there are still a lot of design considerations that you want to have. And, most importantly, looking at demographics, looking at things like that. We also want to look at some of the assumptions, right? Some of the assumptions of your algorithms, assumptions around things like insurance, access to healthcare, and making sure that as best you can, that's represented. And if it's not, it doesn't mean like, oh, you can't do any studies, but you have to account for that, right? You have to, at the very least, you have to talk about that. There are different types of data. You obviously all probably know this. You can have clinical data, which is going to contain lab values. You could have insurance data. You can have electronic health records, which will even have notes. You could have images, any number of things. And then you could have patient source data, right? Things like surveys, Fitbits. We've scraped social media before for certain posts. I know with the Veterans Administration, this is something they've been doing around like suicidal ideation detection sometimes. So there's a lot of different ways to get data about people's personal health information. Let's go back here. So this is a use case that I'm going to talk about at the end if we have time, where we worked with the Office for the National Coordinator for Health Information Technology. So I will abbreviate this to ONC. And so ONC, and we actually worked with NIDDK as well. We had a few partners there to look at predicting mortality in chronic kidney disease patients during the first 90 days of dialysis. There were a few different reasons. This took us 6 months just to get to the question, to be honest, of a panel of experts. But I'm going to blow through all of that and just get straight to the question for you here and say, one of the things we first started doing and how we normed on this question was actually we looked at our potential data sources. And so we ended up pulling from, I believe, 3 or 4 and merging all of that data together to build a machine learning model that could predict whether someone would survive or expire in those first 90 days. So this was a classification model. So we're going to talk about bias now. Let's dig a little deeper here. A lot of different types of bias, a lot of things to account for. Socioeconomic is only one of them, right? We have, social determinants of health, and these biases can start from, like we spoke about, bad design, bad data collection, maybe just poor data for all of the different groups that you're trying to represent. Insurance especially is a big problem because if you have an underinsured population, they may not be represented because they're not even getting healthcare, right? And so, it's not necessarily your job to fix this, but again, it's just something we want to account for and do the very best we can to mitigate that. With chronic kidney disease, this is again, back to my kind of use case I'm giving about chronic kidney disease. We had a bunch of different, and I have a source for this diagram here, a bunch of different things that we needed to think about, including insurance segregation and discrimination in the healthcare system even. And so, digging a little bit deeper into racial bias, it's been there's a lot of data out lately about, women of color being treated very differently, by clinicians and healthcare professionals. And so you can only imagine that this is reflected in the data in ways that we aren't necessarily even aware of, right? So this kind of bias that exists in society that's sort of covert is even going to come up, and not to mention the more overt bias that's a little bit easier to detect, like maybe there's actually a biological reason and there are certain populations that are more prone to certain diseases, right? So you've got sort of biological as well as societal factors to think about when it comes to racial bias, and kidney disease is no stranger to that. So specifically in AI, AI is going to exaggerate these biases if we're not careful. And it's going to, the thing about AI, especially deep learning, is you need a lot of data because it's learning from examples. It's just like a person almost, right? Like you just give it a ton, except it's a lot dumber, honestly. So it's just like a kid, you're going to teach it language, you're going to give it a ton of examples. And like over years of time, it's going to be fluent in English and smart and all of that. And so these neural nets are the same way, except you have to give them a lot more data. If you're giving it a lot of data that's biased, boy, is that going to be reinforced, right? So, we have to be particularly careful when it comes to building these models, releasing them, and, understanding what bias could be there and what the sources are, right? We have things like missing data, sample size, measurement error, right? Sometimes there's actually bias in like certain data collection practices, right? Because people are making an error, or for example, let's say you've got a healthcare system that has 2 categories for gender, and then you've got another healthcare system that has 3 or 4. There's a bias in the one that's binary, right? So there's a lot of places that it might come from. I'm going to let Luca speak to this slide.
Sure. So we wanted to provide you with some examples of the mechanisms that can lead to racial bias here. So we found this diagram particularly helpful. So bias can be data-driven when it results from the data, so from either bias samples or data gaps. And then you can have algorithmic bias resulting from either imbalanced classes or training bias, so from the way the algorithm was trained. And then, of course, you have human bias from societal prejudices and power imbalances. So when the training is done, data-driven bias, algorithmic bias, and human bias can be combined and result in that biased output that we discussed. Next, please. Yeah, so this is kind of the big picture, right? So how biased AI design and deployment practices, which you can see in blue there, that result from discriminatory data are then applied and lead to application injustices. And application injustices, of course, lead to real-world patterns of health inequality and discrimination. And again, the circle continues because the data is going to reflect those inequalities and discriminations. Next, please. So we wanted to provide you with some examples of algorithmic bias, and so real cases, things that happened. So in the first example, an algorithm was used to predict which patients would benefit from extra medical care. The problem was that the algorithm flagged healthier white patients as more at risk than sicker Black patients. And the problem there was that, well, the bias, let me say this first. The bias was discovered when researchers from a health system in Massachusetts found the highest scores in their patient population concentrated in the most affluent suburbs of Boston. So something wasn't right. Next, please. And the cause was that the algorithm used a seemingly race-blind metric. So it was assessing how much patients would cost the healthcare system in the future. But the problem was that the cost isn't a race-neutral measure of healthcare need, because of course there is unequal access to care. So we spent less money caring for Black patients than for white patients. So the solution in this case was that researchers tweaked the algorithm and to make predictions about their future health conditions. And the percentage of Black patients receiving additional help increased from 17% to 46%. Next, please. Another example is the flawed racial adjustments in kidney function estimates. So race forms part of the algorithms used to assess kidney function through an equation, which is called EGFR equation. And this equation uses several factors, including age, sex, and race. And the inclusion of a coefficient for Black patients in this equation was decided based on small poor quality studies. And also the more accurate equation that replaced the first one still contains a correction for Black patients. And the problem is that this equation modifier increases the values for Black individuals by nearly 16%. And this alters guideline-based diagnosis and referrals for care. Next, please. Thank you. So the cause in this case is that including this adjustment ignores that there is a substantial diversity within self-identified Black patients and other racial or ethnic minority groups. So the way this was solved is that healthcare organizations have started removing that adjustment, which is race-based, and they are reporting the white other value for all patients. And this measure is estimated to increase diagnosis among Black adults, so including referrals, increasing referrals as well, referrals to care, to specialist care or other therapies. Next, please. So the third example that we wanted to provide is pretty famous, actually. It's often cited as an example of racial bias. And it's an example where AI-driven dermatology was leaving dark-skinned patients behind. So some programs had been created that were capable of distinguishing between images of benign and malignant moles. And this was supposed to help physicians, help dermatologists diagnose those conditions. But the problem was that an open source repository of skin images from primarily fair-skinned populations was used to train the algorithms. And so as a result, lesions on patients of color were less likely to be diagnosed. So the algorithms provided that diagnosis, but it was less accurate. Next, please. So in this case, bias emanated from unrepresentative training data that reflects, of course, historical inequalities because white people have more access to care. So the solution in this case was to take steps to ensure a more equitable demographic participation in clinical trials and to expand that archive that is being used to train these algorithms to include as many skin types as possible. Next, please. So I hope these examples were helpful, and I'll turn it over to you guys. Thank you so much.
Thank you so much, Luca. That was great. Yeah, so so now how do we, mitigate these challenges? How can we, solve it? And those examples actually included some of those solutions, which is a great one. And we have other things like, so if we have data diversity problem, right? If we don't have the diversity in the data that matches the population, then, step one is to assess those limitations. Step two is to identify a strategy to mitigate that as part of the research design. And I know that doesn't sound very, detailed, but that's because it really depends on the data and it depends on the use case. An over-reliance on machine learning solutions, we mitigate this by including human approach as well, right? We wouldn't just let the algorithms go and be like, well, don't need doctors anymore, right? That's not a good solution. The third one here is algorithms based on bias data. We've been talking about this through all the other slides. You want to collect the best data you possibly can. And then last one is non-clinically meaningful algorithms. This is, a big part of the research design. You want to assess something, you want to try to predict something that is clinically meaningful, right? Like my example of the white blood cell count is kind of a good one of something that's like, okay, and what, right? That's not super clinically meaningful. This is why domain expertise of either your own or working with a domain expert is really important in the research design and in mitigating the bias. So, some say there's a trade-off between fairness and accuracy. Fairness is again just like the reduction of bias. Think of it that way. So is the model being fair by being equal across all of the, let's say, population demographics? And so you could eliminate these sources before training the ML. You probably can't. You may not even know about all of them, right? So step two is you make some fairness adjustments to the algorithm, like Luca was showing, right? They have actually were able to adjust the algorithm to account for the fact that they probably didn't have much data on people of color and didn't have much data on people that were of a lower socioeconomic class, things like that. And then we also have a simple assessment, right? We can adjust the performance after the algorithm too, rather than just tweaking the algorithm, we can do something afterwards as well. There's no real rule of thumb, this is how you do it. There's a little bit of a disagreement. It's still an active area of research. There's a lot of, I've put some, journal articles down here that, you can look into, and, hopefully you'll be part of that research too. So we want to have, so fair and care are really important to this challenge, to all data, especially healthcare data. So we want things that are findable, then also able to be accessed, interoperable. So, and that means that I can use it with multiple algorithms, and reusable. So something that is explained, right? Something that I can use and then I can send it to you and you'll understand it too. The data also needs to be for the collective benefit of those from which the data was collected. And those populations maintain the authority to control the data. These data collectors have a responsibility to interact with minority populations respectfully, and that the ethics of populations from which the data was collected are always being respected. And, this is all to sort of say that, there's a need for transparency and respect. And this will help us to create a better research community and just be better, more ethical researchers producing better, more ethical algorithms.
So I think we
Okay.
have another Slido poll.
Okay, great.
Kevin, can you put it up, please? So if you can go to the your Slido now and answer the question.
Select a potential source of bias for an electronic health record data set. So you're looking at some EHRs and you want to select a potential source of bias here. We have sample size, measurement error, equipment choice, missing context, or all of the above. Since this one has a right or wrong, we'll wait for everybody to chime in here.
We only have 50 people doing it so far. That's way low. It slowed down, so the answer is,
Answers all of the above. So all of these are potential sources of bias. Great job. Almost everybody got it. So you really can't go wrong on this one, because like even if you just selected one, you're still correct.
next.
All right. Thanks, everybody. Okay, next one is the type of model that can be used if you have a set of labeled data. This is data with labels. Unsupervised, supervised, independent, or observation.
You guys can play because nobody knows who's answering the question or not. So it's kind of like just for fun. Okay, and the answer is, Summer?
The answer is supervised learning. Excellent work, excellent work.
And we have one last question.
An AI-ready data set does not need to be fully documented because the model will do it automatically. This one is a bit of a preview into your next section. This is true, false. Yeah, you know it's false.
Yes.
You gotta do that yourself.
Good answer. Okay, next. Next part, we'll go back to presentation.
Yeah, excellent. I think that's it for the slides. I
Yeah.
mean, for the Slido.
Or Slido, yeah.
Quiz. Okay.
Back to the slides.
So let me share again, and I'm actually going to hand it over to, let's see who is next. To Courtney.
Great. Thank you so much, Summer. Okay, so now let's talk about the actual data and what it looks like to actually be doing data handling. So the first thing you're going to need to do is to import your data. So today we're going to be working within a Jupyter Notebook environment. And so the nice thing is you can save your data in the same folder as your notebook. If you're using R, which I will be using for my presentation, then you can use some of the following commands. So these are functions that you just type right into your console. And the first one is getWD, which is get working directory. That's going to print out the current directory that you are in. So either save your data in this location, so the computer will be able to access it, or if you'd like to save in a different location, or if it is saved in a different location, you need to point your R to that location, and you can do that with setWD. And then within that function and within the double quotation marks, you point to the directory that you want to use. Once you're in the directory you want to use, you can check to make sure your data is in that same location with you using the function list.files. And that'll just print out all the files that are also in that directory that you are currently pointed to. So next thing I want, yes.
Excuse me, Courtney. Just for you guys to know, we're going to go over this in an overview. It's not necessarily for you guys to demo right now. We're going to demo after this. So it's for you to see what to do. And by the way, these slides will be made available to everyone. And so as Courtney is saying, you just have to cut and paste it. Then you'll be able to cut and paste it off of the slides once they're posted. So it's kind of more important to really try to pay attention now if you've never done this before. And then when they go into demos, you're going to have a different experience. Okay?
Yes, thank you, Debbie. Exactly. This is kind of a broad overview right now of what you're going to be doing with your data handling, because once we get in and start actually handling the data, a lot of questions and a lot of issues will come up that don't make sense to talk about as an overview, but a lot of data handling does make sense to talk about as an overview. So as I was saying, the data file that you're going to have is going to come in with different types of formats. Some of the really common ones are called comma separated values or .csvs, and Excel spreadsheets are another really common one. And then the rest of them that I have listed, you probably won't see very often, but this is a good chance to look back if you ever do need to open one of these. So some of these are, at least for R, are kind of within the programming environment already. They're built in. So all you have to do is use a function to call them in. Other ones, you have to download a library, which is just an external package of more functions that you can use. We'll be going over that more in the demo, but this is just sort of a handy chart to look back on in case you see some data that you've never seen before. Next. Okay, so like I said, in order to open some of these kinds of things, you'll have to install a package. This is something with, at least within the R environment, you only have to do once. Oops, thank you. And so you run for the first time the function install.packages and in quotation, the package name. Then after that, you need to call the library up, and that time you use library and the package name without quotation. So that's a tricky point that is good to remember. And then you'll need to run the library every time you want to use the library, but you only need to install the package once. Then, so as an example of something like I have an Excel file and it's called data.xlsx, I would need to first install the package OpenXLSX. Then I would need to point R to that library of OpenXLSX as the package, and then I would read in my data. Next. Okay, so now you have read in your data, and I know that's a really complicated step, and it's one that's probably going to take you a while, but let's pretend everything worked very smoothly and we have our data read in. So the first thing we want to do was be sure that we read it in correctly, because the data set may have, the first row may be headers, or it may be on an Excel and it's on a certain sheet within that Excel file, and there's other sheets as well. So we want to make sure that we read it in correctly and how we expect it to look. We also want to get a feel for the size and the complexity of the data set that we're working with. So here are a few functions that you can use. Head takes a look at the first 5 or so rows of the data set. And really you're looking at this to say, does it look how I expected it to look? Dim is a chance to understand the dimensions of the data set, and it will report back to you in rows and columns of your tabular data set. And then finally, names is going to give you the column names of your data set. So these are just some really important first looks at your data. Next. Okay, so the next thing that's really important to understand about your data is that, at least within R, each value was called an element. And data types within R, element types include character, numeric, integer, complex, and logical. You'll very rarely ever need to work with integer or complex within R and within healthcare data, but you're going to obviously be using a lot of numeric and character data. Numeric data is numbers, and they are numbers with or without decimal places. And character is any sort of free text that you might have entered, any sort of labels, any sort of descriptions of your data. And then finally, logical is just true, false. And that's something that we'll use a lot within our coding in order to make subsets of our data or do tests with our data. So we'll be asking sort of tests, and if the answer is true, we will do something. And if the answer is false, we will do something different. Next, thanks. So R can handle more complexity also, not just these individual elements and their labels or their types. R can also handle vectors, matrices, data frames, and lists. So if the most important ones, I'll go over here. So these are all called objects, and they are composed of elements. So a vector is made with the concatenate function, and it's a C with parentheses. And within that, we can have a couple of examples, like a string that has several colors in it, red, yellow, and green, or a string that has several numbers in it. A matrix is made of numeric elements only, and that will come out with rows and columns like a matrix in mathematics. And then finally, a data frame is made of many vectors of the same length, and that sort of looks like a matrix, except you can have all different kinds of information. And that's what your data set is going to read into when you read it into R. It'll likely be as a data.frame. So some really important things that I want you to know about. Why am I going into all of this detail? Because these are the things that are really going to trip you up when you're first reading in your own data set. So the first important thing is all elements of a vector must be of the same type. R uses a process called coercion to attempt to make sense of the input that you have given it. So taking these two things together, if you create a vector like red, 1, and true, R will coerce the elements to be the same type. So in this case, it will force the 1 and the true to also be characters. So what does that matter? Well, it matters because a typo will coerce a vector to become a different type than you expected. So a typo such as 4 point, when you meant to put in 4.0, that's going to be what's going to read in as a character because it's expecting something after that decimal. So now the entire vector will then be coerced to a character. So now it won't behave as a numeric vector when you try to analyze it. You won't be able to find the minimum, the maximum. You won't be able to do math on it. So a really important thing at every step is what class do I expect from this variable? If it's age, I expect it to be numbers. If it is gender, I expect it to be character. If it doesn't look how you expect it to, check for errors and typos. So quickly, let's just pop into a Slido here. So why do you want to use a programming language like R or SAS rather than working within a spreadsheet environment like Excel?
So Kevin, could you put the Slido back up? And for those of you, go back to your Slido link and please put in your answer.
Yeah, reproducibility, that's certainly a good point there, is that we want to make sure that all our data steps are reproducible, and Excel doesn't have an ability to handle reproducibility like writing a code script. It is certainly easier to do statistics and data manipulation with too. I agree to that. Though actually kind of what I'm hoping that you might think about is that Excel is actually very easy to do data manipulation in. If you have your data set open and your cat jumps on your computer, you have now altered your data forever. So that's probably the main thing that I'm thinking about here in this is that these typos that come up that when we read our data into some sort of programming language are very hard to find, are very easy to accomplish in Excel. When you do your data entry and you make a mistake, you accidentally don't continue to key in that 0 after the 4 point, or you accidentally hit an O instead of the 0. Now your entire column has been coerced to a character, and someone who is doing the data handling is going to have to go in and find that error, which is really hard to see within the Excel spreadsheet environment. All of these are also really good answers and are absolutely true. But as someone who has worked with health-related data coming at me from clinicians, I think that probably 90% of my time is spent cleaning up Excel spreadsheets that they could have done a better job of putting in the data in the first place, but it's very hard within the Excel environment to see the things you're doing wrong. And we're going to cover now a lot of the things that end up going wrong within the Excel environment. All right, Summer, we're ready to come back. Wonderful, thank you so much. Okay, so I'm a very visual person. I like to look at pictures much more than I like to look at just these numbers and data points on a screen. So one of the next things that you'll probably want to do to help understand your data is to visualize that data. So a histogram is a picture that's suitable for numeric data with at least theoretically continuous values. So by at least theoretically continuous, I mean height, weight. Those are absolutely continuous things, but we can only measure them maybe to the pound or the half pound or the half inch. So those are coming in with sort of a not a continuous possibility, but we know that they could be continuous. That's also true of age. We might record it just to the year, but we could get down to the 6 months or the 1 month or the day or the hour if we really wanted to. So what a histogram does is creates a specified number of bars, and you can set that represent a value range. Now, the height of the bar is equal to the number of observations within that range. Here, I've just plotted a little example of very simple numbers and the histogram that results from that. So what am I looking at when I'm looking at a histogram? I'm kind of looking to see the sort of the minimum and the maximum that's going on here. This is a really good way if you have a lot of recorded values of a particular variable to see something that might be strange about it. Maybe all of your variables are clustered around 0 through 10, but you have one that's 10,000. That's suspicious. And it definitely makes a histogram that doesn't look kind of within a reasonable range like this one does. Another type of visualization is a bar plot. So a bar plot is suitable for categorical or count data, and it generates bars for each category with the height representing the number of observations within each category. Why is this different from a histogram? Well, because we're not looking for some trend in a change from blue to green to red to yellow. They're not related. We can't stick them together and say that there's any sort of increasing over time. These are separate categories. So in an example with some data here that are of different colors, what I've done is first taken a look at a table that tells me how many counts of each of the classes that I have, and then I'm creating a bar plot of that table. I also tossed in to color the bars to match these colors. That obviously doesn't make sense when we're not talking about things that aren't, where we're talking about things that aren't colors, but in this case we did, so it makes for a pleasing plot. Now, another thing that's really common to see is a scatter plot, and that's suitable when you're looking at the relationship between two continuous variables. So they both have to be continuous, and then we are plotting each number of occurrences of the pairs of the data. So these are most often plotted with the independent variable on the x-axis, and then the dependent variable or the response on the y-axis. So in this case, we're looking at some trees, and they have a height that ranges from about 60 to 90, and the higher they are, the heavier they are. And it would make less sense to talk about the heavier they are, the taller they are. That relationship is also true, but it's probably not that the trees are of a specific weight and therefore grow to a certain height. More likely they grow to a certain height and in doing so gain more weight. Okay, let's talk about another quick Slido quiz right here. Thank you so much, Kevin. So I have 20 observations on a patient's reported gender, sex assigned at birth, height, and weight. The data is in a .xlsx format, so I can open it in Excel. So what do you expect the class in this order, gender, sex, height, and weight to be? Do you expect them to be category, category, number, and number, factor, factor, integer, and integer, or character, character, numeric, numeric? Yes, awesome. Everybody got that one. So yes, we haven't talked about category or number yet, and we haven't talked about factors yet. So the factors could be something that's true, we haven't talked about it yet, and it's probably something that we would have to accomplish within the programming environment and not something that came natively with the data set. Okay, continuing on, which of the two variables can I visualize together using a scatter plot? Gender and sex, sex and weight, or height and weight? Yes, exactly. We all got that one. So height and weight are two continuous categories, and therefore we could have lots of different combinations of height and weight, but we wouldn't see very many combinations of gender and sex. And we would see probably a few combinations of sex and weight, but that's a different kind of visualization than a scatter plot. And then finally, imagine, this is more open-ended, just kind of think about what we've talked about so far. But in R, I ran hist weight to create a histogram of weight, and I received the following error. What I see is error in hist.default data money weight : X must be numeric. That looks kind of scary, but what do you think is wrong and what could I do to investigate? What within that kind of strange input do you see that you can kind of grab onto and take a look at? Yeah, so I see a couple of really good selections, responses here. So as.numeric is something that could coerce it to numeric if that is possible. If there's something in there like female or red, it's not going to be able to coerce that to numeric. But yeah, for sure, something is going on here and it is not numeric when we expect it to be numeric. And yes, it definitely, we could think of looking for a typo. We don't know what it is. So yes, it certainly may be that there is just a decimal place without anything at the end, but it could be something else entirely. Yes, some people are definitely jumping ahead with the kind of nested number of functions there. Yeah, you're right. There is lots we can do about it, and we'll talk about that a bit more.
All right, I believe that was the last question of this.
Yeah,
lot,
that's
so
great. Thank you so
talk-
much. Okay, Summer, back to you. Okay, this is great. Now we're thinking a lot about not only the data that we have, but these individual elements and what it looks like to actually clean them and handle them. So one of the big things you're going to find and I'm sort of imagining that this is data that you've inherited and that you are the data analyst in this situation. But if it's data that you have collected, this is a really good way to think about how to collect good data. And that is something that is really hard to teach and really hard to toss in a curriculum, because what do you have to know in order to collect good data? Well, you have to know how to handle data. How can you handle data if you haven't collected data yet? Kind of a catch-22. So all the time that I'm telling you different things that you can do with data you've inherited, imagine this is data you're collecting instead, and be a wonderful data collector. So one of the things that will slow down and anger you quite a bit is typos. So a common place we see typos is in self-identified categories. So sometimes data sets will use codes rather than the actual labels, so that these variables look and behave like numeric count data instead of categorical data. So in an effort to try to standardize data collection, people may input race as 1 equals white, 2 equals black, 3 equals Hispanic or Latino, 4 equals other. And that sounds like a really good way to standardize your data. And for the most part it is, but it also allows you to do some crazy things like find the mean of race. And that doesn't mean anything. So instead, R gives you an opportunity to handle factors so that you can continue to use the categorical names, but they will just be treated as labels with underlying numbers underneath, and it won't allow you to do things like take the mean of race. But another thing to really look out for and pay attention to is if some of these values, if we used the values instead of numerics. One thing I will say, if you use numerics, it's really easy to make typos and you will never see you did it wrong. You don't have anything to go back to say that that person actually self-identified as Hispanic and you accidentally keyed in a 2 instead of a 3. That's just in there forever. But instead, perhaps you input your data using actual labeling and words. But then we tend to see a lot of typos and a lot of inconsistencies for now, where we might see use of white, all lowercase, white with an uppercase W, white, all caps. Someone else may put in HISP as a shortening for Hispanic, or Latina because the person is a woman, all kinds of differences here. So the first thing that you can do is take a look at the table function, which is very helpful because it works on both numeric data and character data, and will give you all of the potential options that were within the column of this data. And then also we will get into a bit about recoding data, about changing the entries, and recoding these isn't too hard. What you should do is pick a standard and stick to it. And we recommend referring to some sort of standard ontology like SNOMED for that interoperability between data sets. So these typos may look like clinically implausible values also. They might look like extreme outliers. So assessing min and max for numeric values is a good way to find these things. So some examples might be an age of 250 instead of 25 because a 0 was accidentally typed in, or a height of 5.4 centimeters because the person was thinking of feet. So these are kind of things that are now getting into not just good data handling, but also putting in your own clinical, your own public health knowledge to understand what could have gone wrong here. Be a detective and find out what maybe has gone wrong. If it's something that's pretty easy to see, then you can go ahead and change it. If it's not something that's easy to see and it looks like it's really out there, then it's not for you to change. And you might want to just take out that value and put it in as a missingness instead. So when handling medical data, you'll probably want to speak with a clinician to understand the clinically plausible values of laboratory measurements, as well as to understand what the laboratory cutoff values may be. So I have had the experience before where I was looking at laboratory values for veterinary situation, and I'm definitely not a veterinarian. So I don't understand why they would take these types of measurements. And I certainly didn't understand why many of them were 1,000, except many more of them were 2,000. And it turned out that they were using two different labs to run these results, and one cut off at 1,000 and one cut off at 2,000. So that's the kind of situation where I really needed to go back and talk to the veterinarian about what was going on there and what should I change these values to be what was clinically meaningful. Okay, so now I mentioned a little bit about missingness. There's actually 3 different kinds of missingness, which maybe you had not thought of before, but missing can occur completely at random. So the fact that the value is missing is independent of the data value itself. So there's no systematic difference between those with missingness and those without missingness, such as a batch of laboratory samples that were processed improperly. So something like that is something that would happen to people, whether they were cases or they were non-cases. There's also missing at random. So missingness is systematically related to something you have observed, such as male patients being less likely to answer survey questions about depression. So here you can do something with that because you've also recorded what is linked to the missingness. And then finally, missingness not at random is very similar to that MAR above, except it's related to something that we didn't observe. So such as not answering the survey about depression based on the severity of your depression. Now we don't know why you didn't answer because you didn't answer it and it was to do with the depression itself. So handling missingness really depends on the type of missingness that you are observing. Okay, so data cleaning and pre-processing, these are the kinds of steps that you're going to need to do with your data. So one of the things we can do is regroup categories. So say we had collected our race and ethnicity in 2 different categories, which is true in census data. And so, for example, we have white, Black, Asian, and other for race categories and 2 options for ethnicity. We could combine these and then we would end up getting lots more nuance in our data. But now it's up to you to assess whether this regrouping is better. You're the expert in the data that you are collecting. You're the expert perhaps in the population you are studying. So it's up to you to kind of add in a little bit about whether this makes sense to do something like this. How do you think that your participants are going to self-report their race and ethnicity? But this does allow us to do some statistical tests to understand whether the missingness is the same in both categories, whether there's some sort of systematic missingness, and whether it's based on other variables that we have collected along the way. Okay, so now handling missingness. So as Summer touched on before, AI applications really need complete data sets for the most part, but health data we know comes with a lot of missingness. So one of the common approaches that you'll find on the internet would be to handle missingness by deleting rows and columns that contain that missingness. But we are healthcare experts. We know how precious every data point is. So the idea of deleting entire people or deleting entire things that we work to collect data on makes me kind of sick. So I try to avoid doing that when possible, but sometimes it is just a necessary thing to do. One thing is when deleting rows that are observations or patients, ensure that those with the missingness are not systematically different from those with the complete information, and you can do that with statistical tests. Also, if possible, maybe you can create a new variable that indicates the missing data such that the model can be fitted with this new variable. And since it is complete, because you are saying, is it missing, yes or no, and that is complete, the model can be fit with that kind of information. And then you can just sort of omit the column that has the missingness out of that model, but you're not losing those actual measurements there. And then finally, if you're going to be deleting columns or rows, use some sort of systematic rule such that the column must contain at least 5% filled cells, something like that, in order to decide to keep it or remove it. Something that is a consistent rule you'll use throughout your data handling and that you document. Okay, another way that you can start to be a little creative in your thinking about handling missingness is if perhaps you see columns like this. The people were asked, do you feel anxious about your diagnosis? Yes or no? Do you feel sad? Do you feel accepting? And maybe what a lot of patients did was maybe they were given checkboxes and they were filling out themselves, or the data handler was filling it out themselves. And they just asked, how do you feel about your diagnosis? And the person said they feel sad. The response was to check that box and to not check any of the rest of the boxes. That's going to cause a lot of missingness within your data here indicated by NA, that it does not exist. And you can recombine these into a new variable in which you can fill in every answer for every person. And then finally, you've probably heard of something called imputation. So that's a method for predicting values based on the surrounding data. And this actually works for all types of missingness that we've discussed, but involves creating some statistical predictive models. So I'm not going to speak to it at this point, but just know that it is out there, and they are statistical processes that are not too hard to accomplish, and something that you should be able to handle after you've gotten a pretty good grounding of basic statistics and data handling. Okay, time to pop out for one more Slido quiz. So here I have, going back to that histogram weight idea, my code didn't run, and I'm interested to know why. So when I run the code and I see the following, one of them is exactly that error that we talked about, the 136 point with nothing following it, that coerced the entire string into being character instead of numeric. So what could we do to handle this? Yeah, sure. I like that answer a lot. That's definitely what happens sometimes in Excel. Doesn't want to give you that 136.0. It cuts off that 136. And maybe that was something that the person who put in the data, they put in 136 point and couldn't see that teeny tiny little point there. You could make the string numeric. Since the one observation is 136 point, then that one will turn into an NA, because it is not something that R understands to make into a number. So you would lose that observation. I would say that this observation at 136 point looks pretty clearly to be a typo and a reasonable weight. So replacing that value with 136 probably would be okay. But if instead it looked like it something like 70, you might not want to make that kind of same, jump to conclusions that this was a valid number. So yeah, recoding it as missing, recoding it as an actual variable. These are all kinds of things we can do. Okay, a lot of things that you'll need to handle also are going to be date objects. Fortunately, at least within R, these are really easy to work with. So with R can handle with the as date function, lots of different formats of dates. You just tell it what the format is supposed to look like, and it can run this and standardize it over your entire column. So sometimes they may be separated into day, month, and year, and you will have to do a little bit of work to combine those three columns before you can use the as date. And so quickly in that 3-column format idea, a function called paste can just glue together each of the entries that you're looking for, and you can specify what the separation is that you want. So that's pretty handy for the as.date, because you can use a slash to separate day, month, year, just like we're pretty used to reading those. And then you can use the as.date. This is an opportunity for me to pause and tell you a little bit about code experimentation. As a good seasoned programmer, you're going to want to do little experiments within your machine. At this point, you would want to look to make sure that you have your code does what you want it to do, that the date coming out of that paste is what you expected it to be, and using the format specifications that you expect it to be. I definitely do not have these date formats memorized. I really just play around in my machine to make sure that it comes out how I intended it to come out. Okay, another thing to talk about is creating new variables. So this is going to be important sometimes in certain types of machine learning applications. And it's really important to understand kind of what R is doing for you under the hood that you don't get to look at, because in R, this is pretty easy. So say I have a list of variables that can be white, blue, red, or black as possible outcomes. What I can do is convert these into 3 separate variables that are indicator variables that say whether or not each application is a part of this group. So the first one, white, is it the color blue? No. Therefore, I put 0 for no. Is it the color red? No, so 0. Is it the color black? No, so 0. So that means basically what I'm saying is I have 4 different potential categories, and I'm going to create 3 new variables, because one of them will always be indicated by not being a part of any of the other variables. Okay, and then finally, important thing to understand is merging files. So there's 2 different ways to merge files. Maybe your data comes from a database and it comes in separate database files. So the merge function is a really easy way to do this in base R. Another version is from the dplyr package, and it has a join family of functions. If you want to get a little more technical into the SQL database syntax, you can merge your 2 different data files in different ways by only pulling those pieces from one file that match in the other file, or only pulling those that overlap in both files, or by pulling the entire thing. And then finally, harmonization and fusion. So data harmonization is to do with creating sort of a larger data set than you originally had by combining several data sets. And so this may occur using census data that is collected every several years, or maybe a clinical trial that was collected over several locations under the same study design or a similar study design. So data harmonization, the data sets should be very, very similar in order to do this. And probably what you're going to need to do is maybe change some column names so that the column names match, or you might need to change some of the label coding, that type of thing, so that everything matches. But otherwise, you're not needing to work too hard to interpret 2 different types of data sets. They should have been collected under similar methods and collecting similar data. When you're combining several studies over several years or locations, it's a really good idea to create a new column that captures that year or that location, so you can also go back and study the original data sets. And then finally, data fusion is a process of combining multiple data sets to test hypotheses and find patterns that would not be testable in a single available data set. So this, of course, when you add multiple data sets and data types, you're multiplying your opportunities for more bias. One of the big important biases here is the ecological fallacy, which you might have heard under many different names, as many different fields understand that when we make inferences about individuals based on aggregate data from a group, that is inferring things about a person that may not be true. AI is very good at committing this particular one. It can infer race based on your home address and your neighborhood characteristics. It can infer your sexuality based on social media likes. AI is made to find these kinds of patterns. So especially with those particular examples, if you just omit the overt categories of race and sexuality, don't think that AI still can't see those patterns. And now it actually can't even work with your real data of the race and sexuality that you actually identified. So in many ways, you're probably making things worse. Another bias to really pay attention to is reusing data sets that weren't collected for research purposes. So maybe you go out on the web and you scrape some data that was collected via cell phones or social media. But we know that these already have their own selection biases regarding age, whether the person is living in an urban or a rural setting and socioeconomic status. So now you're just bringing in bias. So also be careful about drawing causal conclusions from fused data. And just to let you know, causal data fusion is a branch of computational epidemiology with a growing body of theory. So you can draw causal conclusions from data fusion efforts, but you need to understand how the data was collected, who the data is studying, and whether or not your combined data sets can answer that question. And finally, very, very important, now that you understand what it means to handle so much data, you need to document all along while you were doing this. So while creating and working with the data set, you're going to also create an accompanying data dictionary. So data dictionary is the documentation of the data set itself, the source of the data, inclusion, exclusion criteria, tell me about the study population, the source versus the target, the sampling schema you used. When you are collecting data, this is documentation of your study. When you are reusing data, this is you interpreting perhaps where you got this data from, where it came from. Tell me as much about it as you can and make sure it's attached to your data. And then also data dictionaries typically are better known for documenting the data elements. So this needs to include thorough descriptions of what was collected, why and how, as much as you can know that, the standardization that you're using, whether you have coded 0 for no and 1 for yes, whether you've used pounds or kilograms, summary statistics for each of your elements, what you use to note missingness, and understanding each of the pre-processing steps that were taken. Really, the goal here is for you to put your data set out into the world and no one ever needs to call you and ask any questions because all of your documentation is right there.
And just to close off Courtney's portion of the presentation, we're going to quickly cover data models, and this specifically applies to whether if you're working with data that's within a database. And so common data models are a means to organize data that's stored in a database into a standard structure in order to facilitate data interoperability with other systems. And so what common data models do is they provide a common format as well as common representation of the variables and other components in the data, such as standardized terminologies and coding schemes to, again, facilitate the standardization of that data. And an example of a common data model that we wanted to show here is the OMOP common data model. That's the Observational Medical Outcomes Partnership Model. And this is an open community data standard that standardizes the structure and content specifically of observational data to facilitate the efficient downstream analysis of that data. And so this is just one of the technologies that you can implement to ensure that your data is fair, specifically if you are working with data that's within a database. I think now we can switch to the demo.
And I just want to add that it's 4:15 now, and I know that some of you have to drop off at 4:30, but as we go through the demo, if you can stay past 4:30, our guest speakers and we will stay with you and bring you through all of the demo. For those of you who need to leave, if you want to catch up with it later, it will be posted on the NIMHD Think-a-Thon portal webpage. So you can always go there and review it as well. So I guess, Courtney, you're going to do this? Courtney and Luca?
Actually do the first part to make sure that everyone has access now, and everyone should. So let me share my slides with you for just 1 minute. Okay, so I would invite everyone to use the link that you see now, which is different from the one that you saw before. So please make sure you have the updated link. And actually, Kevin and Elizabeth, can you please paste it in the chat?
Yes,
So
it's been
please-
pasted in the chat by Elizabeth.
Thank you so much. So when you do that, now everyone should be able to skip this part and just see this screen, because you should all have access. Okay, so if somebody doesn't see this screen, please let me know, send me a direct message. But you should be able to see. And then the next step is going to be preparing the virtual machine that is going to run the computations today and setting it up. So please click on environment configuration. It's that button with a cloud.
So I'm just going to give you a little pre-warning. When you click on that button, you might get an error message that says loading files message. Some of you have already put it in the chat. What that is simply, it's synchronizing, and there's so many people trying to do it at one time. We just hit it again and it will in time do it. Just, it's almost like you're in a queue, kind of popping it up. So don't panic if you get an error message.
It's just a matter of retrying in a moment.
Yeah, just retry it. It's just so many people. It's like, you know, in the olden days when Gladys was on the other end of the phone and she had to patch you through, that it's kind of the same.
So if you click on that button, then you should see this screen. It says Cloud Environment Details. And you have to click on Settings under Jupyter. So please do that. Click on Settings under Jupyter.
Give it just a second, Luca, because they're still synchronizing. Says edit is disabled. What does that mean, Luca? Some people are getting edit disabled instead of an error message.
Okay, let me do one thing real quick. I'll stop sharing for a moment and I try to fix this.
For those of you who were where Luca is at, it'll help if you go ahead and press that Jupyter Notebook signal so that it can get processing and starting to upload. Because remember what Courtney said is that you have to download these notebooks in order to actually start running the program. So that is what we're doing right now. We're doing the download of the programs so that you can actually run the code. And so she showed it to you on a slide, and you're actually going through that experience right now. We'll get to what data set to download. Right now, all you have to download is Jupyter Notebooks on the setting.
So
Or Studio.
We should be ready. Oh, sorry, Debbie. You shouldn't have the problem anymore. Can you confirm that, please?
Can everyone please try hitting?
Yeah, maybe refresh the page.
Refresh the page and try again, because hopefully Luca was able to fix whatever the edit problem was. And then are we using Jupyter or RStudio?
We're using Jupyter.
Okay, so
Jupyter support Python.
you can click on the Jupyter setting, click on it.
Yeah, right there.
And by the way, the reason I asked the question, some of you will already know, because Jupyter is more for Python and RStudio is for R language.
Right. So hopefully the problem is solved. And It's from the chat. It looks like it's fine.
Nope.
And
It looked like Penny's.
yeah, it's still
porridge.
processing. Yeah, it's gonna work. Don't worry, it just takes time, but it was a problem with permissions.
So please go ahead and try clicking it again. Looks like it still has edit disabled, Luca.
yeah, it's gonna take time. So you see, for some users, it already works. It's the same issue where when multiple users are trying to do the same thing at the same time. And you do need to refresh your browser. Yes, thank you, Troy.
Could you back up one step, Luca?
Yeah, of course. And Aisha, what to click in analysis, I'll tell you in a moment.
Okay, but back up one step, Luca. Yeah.
Of course. So here we are. Please click on environment configuration on the right.
The little cloud thing to the right of your screen.
And when you click on that, it's going to show this screen. So here under Jupyter, click on settings. I'm going to say this again later, but just going to go to the next step, which is actually modify the settings. So in this window here, don't worry about anything. Leave the default values as they are and just scroll to the bottom where you will find either a create button or an update button in the same location. It's update for me because I've already done this in the past many times, but it could be create for you because it's the first time. Click on that button, leaving all the values unchanged.
So some of you who've been in our workshops before, it could say update, but mostly it's going to say create. So just hit the button that says create.
Exactly. And then I'm going to go over the steps again, but I just want to show you while we wait, because creating a virtual machine takes 3 to 5 minutes. So we're going to have a poll in the meantime. We're going to use that time. But I'm just going to show you what's next. And then I repeat these instructions. So I have a congratulations message here. And then in the analysis, and this is to answer your question, Anisha. So you have to find notebook number 9. And I'm going to show you, but there are multiple copies. So I would invite everyone to click on the one based on your last name initials. I'll show that in a moment. But that's going to be the next step. Actually, I'm going to do it right now. I'm going to show you.
For those of you who are still getting the disabled errors, please refresh your browser and then go back out and try again.
Yes, and I'm going to repeat the instructions again. So basically, you see that there are multiple notebooks number 9. And the title says last names initials, for example, A to C, right? And then D to H, I to M, N to R, and S to Z. We did this to avoid, having multiple users working on the notebooks at the same time to avoid lagging issues. So please click on the one that corresponds to your last name initial. They're all identical. And I'm going to repeat the steps one last time, and then I'll turn it over to Courtney or Shelley. Courtney.
We still have edit error messages.
It's normal, unfortunately. It's going to take some time. There are like 100 users right now trying to do the same things at the same time. You just have to be patient and retry. Refresh the page. That's important. Refresh the page. and retry. Worst case scenario, you can log out and log in again.
And I love that if you find something that works, that you're putting it in the chat. Thank you.
So the steps, once again, are paste this address in the browser. You will see this page. In this page, click on the environment configuration button with the cloud on it. Then click on Jupyter settings. And it's going to bring you to this screen. You can leave all the default values unchanged. Just scroll to the bottom, click on the create or update either button, and you should be good. And now you have to wait about 3 to 5 minutes. We're going to do a poll shortly during that time. And when we are ready, when the environment is ready, it's going to say environment ready. There's going to be a message on the page. And then I will ask you to go to that list of notebooks, identify the one that corresponds to your last name initial, and click on it. So I'm going to do it myself. I'll show you. You just have to click on it. And then click on open.
So for everyone, that the notebook that says preparing AI ready data sets, it's the same.
Yeah.
But what we found is that, as you can see, with hundreds of people trying to get it, it creates too much delay. So it's exactly the same notebook. So don't worry that somebody's getting something that's not for you. So we created it by your last name. So click on the notebook that goes with your last name. It's all the same.
Yeah. I'll just click on one to show you. Click on the first one here. And then click on open. I'm going to do that now. Yeah, you see I'm running to the same problem, right? Because there are too many people doing this. So it says starting cloud environment. This may take up to 2 minutes.
So for those of you that are having difficulty, you can just watch. And again, this will be posted on our webpage for you to do it at your own pace. Troy has a nice suggestion. Some can go S to Z because there's fewer people with that name.
Mm-hmm.
So that's another option. But we're going to kind of move along now because we're only have 3 minutes left, and I don't want to keep all of you more overtime than we need to. And for those of you who can stay on, please do, and we'll walk through this and give you some hands-on experience. But again, it will be posted on our webpage. And Elizabeth posted the link where you can go.
Perfect. And as a reminder, the next step is to click on open here. Okay, I think we can do the poll right now while we wait. I stop sharing. I would ask Kevin to please.
Kevin, are you bringing up the poll?
Bring up the poll.
Yes, it should be showing now. Sorry for the delay.
The question is, properly handling self-reported demographic data is an emerging field of interest. What are your thoughts? Debbie, would you like to comment on the answers when they show up?
Well, we both can. What we're trying to get out with this is some of the challenges that we have in health disparities research is self-report and the amount of data that we ask. There's still, and for those of you guys who were following along in the chat, there was a big debate about race being a biological nature versus nurture factor. There were several comments about there are biological differences, and then there was other comments that it's just a sociological factor. So these are the kinds of things that make it very difficult in getting conducting health disparities research when certain demographic variables are not collected or the way that they are collected, because self-report is one way, but sometimes people just look at you and say, oh, I think you're, and they assign a race and ethnicity. So this is some of the challenges that we have with this, and also why so many biases occur in AI types of analyses, because as both Summer and Courtney and Anya have addressed today, when a machine is either self-learning or we're putting in data, it makes it very difficult then for you to get information out that is not biased. The reason that some of these polls are also very important is because we use this information to help tailor what we cover in our Think-a-Thons. And so the collection of race and ethnicity or data cleaning, there was lots of comments in the chat about synthetic data or imputed data for missing race and ethnicity variables. So these are all very important topics for us. So please share your ideas.
Debbie, let me know if you want me to move on at some point to the closeout survey as well.
Actually, since we're in Slido, let's just go to the closeout survey, Kevin. And for those of you, that doesn't mean we're closing out. We're going to do the demo. But for those of you that need to leave, we'd like to do the closeout survey, and then we'll go back to the demo, okay? Go ahead, Kevin.
Absolutely. So it's launched in Slido. So if you're in Slido, you should be able to see it. We obviously will not display results publicly for this survey.
And by the way, we're going to leave this up and you can fill it out, this survey out. This is our evaluation survey, and please do so because we try to make these as friendly as possible and as useful as possible. So please give us your feedback so that we can do a better job. And I'm going to turn it back to Luca and the demo.
Of course. Actually, I think, Courtney, would you like to take over
Courtney?
with the demo?
Yes, yes.
I'm
Let
here
me
to
share my
help
screen.
in case any issues arise. But I think everyone should have been able to open the notebook.
And if you didn't get to open the notebook, just watch along so that we can go through it and minimize the overtime. So Courtney, if you want to start.
Yes, yes, I am firing up.
Thank you.
Okay, hopefully we can all see this. And once you finally get into your workbook, this is what it's going to look like. So when you do get into the workbook, if I could ask you just to scroll down to these couple of cells here, and you're going to click on the cell, and then in the upper left where it says run with a black triangle, go ahead and hit that because it's going to take a little while to run. And then the next one, also this install packages, OpenXLX, go ahead and run that one too. That way, while I'm talking, they will be running in your background. Okay, so
I'm sorry, Courtney. One of the things for the new users, if Courtney, can you go back to those two, please?
well, I just need them to run
Yeah,
in the
I
background,
know.
and I will talk about them when they get there.
I know.
Okay.
For those of you guys, this is important parts that we use in the notebooks. You don't have to know code to do this. You can just go to these links and run it. So this is kind of, the notebooks are important for that ease. So Courtney, go ahead.
Great, thanks. Okay, so what I'm going to cover is the same things that I covered in the slides. We're going to talk about importing data, viewing the data set. We're going to talk about element types and object types. We're going to do some visualizations, look for typos and missingness. We're going to handle some dates. We're going to create new variables and factors. We're going to do some renaming. We're going to change values to consistent names. And finally, we'll touch on merging, harmonization, and fusion. So starting out, importing, viewing, and installing your packages. First, the thing we need to do is import the data that we'll be working with today. So this is a code cell, and I'll tell you that the previous stuff was called a markdown cell. So what I can do here is open this up. This is how I created this. I created this with a cell. Jupyter notebooks are all loaded up into individual cells. And I took this cell here and I used this drop-down menu to call it a markdown cell. And then when I had it all created how I would like it to be using markdown language, then I hit run. And so then it runs and it shows how I want it to show. So that's how I created this notebook in the first place. And that's how you are going to want to work with your notebooks as well. You'll want to intersperse code cells along with documentation cells. And the nice thing about a Jupyter notebook is you can do both at the same time. In order to import this data that we'll be working with today, you just click on this cell and you hit run. And what happens is right here, it has a line number for you, and that line will be an asterisk while it is running, and it will update to the numbered cell when it is done running. So what we've done here is a little bit of code, like Debbie said, that you don't need to understand at all. It's what's going to get you access into the data set for this example. So as discussed previously, data sets can come in many different formats, and we need to tell sometimes that they are able to be read in natively in your programming language environment, and sometimes you need to install a package in order to do that. So specifically today, the data set that I have created, and this is a synthetic created data set just for us to play with right now. Specifically, I made it as an Excel file so that you would have to practice installing libraries and using them. So the package that you need to use in order to open an Excel file, the one I prefer is OpenXLSX. So to install a package, you put that package name in double quotes, and then go ahead and run that cell. It will take a little while to run. So if you're following along with this demo, hopefully it is already loaded up for you by now, and it gives you this little message that it has installed and lets you know that it is there. You also know it is run because the line has a number. Okay, so that's a step you only have to do once, fortunately, because once it is installed in your instance, you always have access to it. But every time that you run this code script, you'll always need to point R towards that library. You do that by writing library and then putting the name of the package you're looking to use in parentheses. So we can run that code now, and then that will allow us to read in our data set. And we read in the data set with the function read.xlsx within this OpenXLSX library. And now I should say that R is an object-oriented language, which means that everything you do, you can save as an object and give it a name. So I'm going to read in this students' mental health data set, and I'm going to name it students. So I will now run that code. That goes really fast. That asterisk turned into a number right away, and we're ready to move on. Something that I like to do is separate out the install.packages code cell from the cells I'm going to run a lot to remind me not to run that install packages again. The other thing you could do is just delete it, but what I like to do is pop up here where it's currently code and just switch it to raw. And that way it won't run again. I can hit run and it does nothing. So it still reminds me it's there. I can go back and use it if I need to use it, but I can just click through all my cells and it won't run. Okay, so first let's take a look at our data by hitting the head, and head is just going to give us the first several rows of our data. So here we see we have names like choose your gender, age, what is your course, your current year of study, your collegiate GPA, and we see some of the first responses for our first 6 participants here. So far everything looks good. This is what I would expect my data set to look like. So I'm pretty happy with that. There's variable names and they appear to match the recorded values. So I could take a look at this and say to myself, what do I expect as the class for each of these variables? And yes, I realized it does say it right there, but let's not look. Let's pretend that it didn't say that. Okay, so another way that you can get a summary understanding of your data set is with the ScHARe function, STR. It's giving you the structure of the data set. And that is going to, when you click on that and run, you'll see that our data set is, or our students is a data frame. It has 105 observations and 15 variables. So its rows are 105 and its columns are 15. We see each of the variables that we saw before in the head, and it gives a little summary of them. It says choose your gender as a character vector, and it's filled with female, male, male, female. It doesn't tell us all of them, it just tells us the first 4 or 5 or so that it can fit on the line. But just like the head, it sort of gives you a sense of what the data is that you have here. And I'll say that there are different settings in the original reading in of the data file. So if this doesn't look like this when you read in your own data, it may be because you need to point to an Excel sheet. It may mean that you need to say that header equals true or header equals false based on the way that the data was encoded. Most often R understands how to read in your data set and that the first column will be headers, but in Excel, people like to get creative and put lots of different headers and merged cells and things like that in there, and R doesn't necessarily know how to handle that. So you may have to play around and reread in your data several times in order to start looking like what you expect it to look like. Okay, so taking a look at this, not all of these variables are what I expected them to look like. Specifically the age. I expect the age to be numbers. That's how I think of age. But right now, all these values are in quotation marks, and this is listed as a character. So this is sort of an indication to me that something might be wrong. And also best practices are to take a look at each of your variables and understand what's going on. In a very large AI data set, that may not be true, but in health data, you usually can take a look at each of your variables independently. So one of the ways that we can explore this is through the use of the table and the hist function. For histogram of age, since I expect it to be a numeric, I should be able to run a histogram on that. However, when I run that, I get an error. And this is the error that we talked about in the discussion section, where it actually points you to exactly what the problem is, that X must be numeric, and we did see that it is character. It also gives you a little bit of debugging. So what's happening under the hood when we use this hist function is it's actually following through lots of different computer steps. The first thing it does is read to make sure that what you typed in was grammatically correct. If instead I happen to have not said students, but I said student instead, when I go to run it, I get a different error because the first thing R did was check to make sure that my input made grammatical sense. In this case, computer grammatical sense. And it did not. It says student does not exist. So that was the end of that. It could evaluate no further. But if I put that S back again and I rerun that, it's able to first read the command and say, yes, that makes grammatical sense. And then it attempts to use the default version of making a histogram. And that's where it no longer could work anymore. It stopped because X must be numeric. Okay. So this error message is very informative because it's repeating what we observed in the structure function. Age is not numeric. So how do we solve this problem? Well, one thing that we can do if the string, if the vector is not too long, is we can we can take a look at it directly. If it is too long, we can also take a subset of values and take a look at that. So you can feel free to read my comments there in order to how to do that, but we will talk about subsetting a bit later. So this one's pretty short, and this is what students' money age looks like. I like to think of these or I should say that in a data frame, each of your columns had names, and you call just the name using this $, which I like to think of as like an apostrophe S. So it's students' age is a good way to remember it. And when we take a look at that, we see that there are lots of numbers. There's a missing value here recorded as NA, but down here we also see one of these values as female. So that's what did it. It definitely coerced the entire string into a character. Another thing you can do to understand what's happening is to run the table. So if your string is long and has many like categorical variables, this is kind of great because it'll summarize it for you. If the values are truly continuous, this is not great. It's going to give you a lot of output, but that's not hurting you. It'll help you just understand what your data is doing. In this case, we see that our students are mostly ages 18 to 24, except one of them is age female. So then what we'd like to do is clean that up because age female doesn't make any sense. We need to make some decisions about what we're going to do with that. At least since this is the very first column we've taken a look at, it might indicate something's actually wrong with the data set itself. So let's find who was age female. We can do that with the which function. And this is doing a logical test here. It's saying for all students' age, which ones equal female? This is a logical test, not a mathematical function. And so we need to use double equals. And then here's another common error that tends to happen. I forgot that they were using that the above answer was female with a capital F instead of female with a lowercase f. So I didn't get the output that I expected when I ran that one because it doesn't have anything that is female. So I just went ahead and adjusted my code. And now I can run that. And it is observation number 102. So we can call individual values or entire rows and columns using bracket notation. For a one-dimensional object like a vector, the elements are all sequentially numbered. And here's an example that I mentioned before in sort of the lecture side of things about doing a little code experiment. So to understand what these brackets do, let's write a little code experiment. We'll create a test vector of different colors, and I can clearly see what each, the first observation is red, the second is orange, the third is yellow, so I can see exactly what I want, right? And I would like the third one. I expect that to be yellow. Yes, the third one is yellow. So that's how that bracket notation works on a vector. For a two-dimensional object like a matrix or a data frame, though that same bracket notation is going to call first the rows and then the columns. So coming back up a little bit, the which student's money age is female was observation number 102. So now I'd like to just look at observation 102. So click on this cell right here, and we will see observation 102. And observation 102 has a choose your gender that is missing, an age that is female, a course that is 22, a current year of study that is law, their GPA is year 3, their marital status is 3.0 to 3.49. So it looks to me like this column has been off shifted. Like everything, like an NA was put in the first and all the rest of the data entries were off shifted by 1. And I don't know why this might have happened, but it could be something like in data collection where somebody was keying this into an Excel or some other type of spreadsheet, and they accidentally started on the next column down from where they intended to. So another sort of tip here to think about is to create a plan to address the issue and then record your plan within your code comments. So here I see that the responses appear to be off shifted to the right. So my plan is to put the first 2 through the last entries first and then append the first entry at the end. So I'm going to use that bracket notation along with a function called length. And I'm going to create a new variable because R is object oriented. I can give this idea of the length of students 102, the length of their data. I can give it a name and I'm going to call it end. And then I'm going to concatenate 2 through the end. This colon here means the numbers 2 through whatever the value end is. So if it was 1 through 5, it would be 1, 2, 3, 4, 5. And then I'm going to stick 1 at the end. And that's exactly what I wanted it to do. It goes 2, 3, 4 through 15, which is the number of columns we have, and then sticks 1 on the end. So now, since I have done a little code test to make sure that that line of code with the concatenate there does what I intend it to do, now I can put it into my bracket notation. So I want the 102nd observation of students, and I want lines 2 through 15 followed by 1. And then I'd like to reprint out what that looks like. Great. So except for the column names, the data now looks in the place that I want it to look like. So we remembered that the first thing they asked us was actually the gender. The next thing they asked was the person's age. And now our data, our actual values we've recorded are correct, just the column names are wrong. But the column names don't matter, because what we're going to do next is merge that up to the original data set, and it'll grab those column names off the original data set. So we're going to take our adjusted students 102, the second observation, with the adjusted valuables, and we're going to assign that. That's what this less than minus symbol here is. It's like a little arrow you made. We're going to assign it to the original 102nd. We're going to replace it. And now when we take a look at it, we will see that everything looks how we want it to. Yeah, so the column names updated when we replaced the original data set. And now we see for the 102nd observation, her gender is female, her age is 22, and all her data sets line up quite well. The only thing that we had was the NA value that we stuck on the end, and that was for her sort of ID, her student ID. So if we take a look at everybody else's student IDs using students ID, we can see all of the values. And except for hers, which is an NA, everybody else's looks like it starts out with 103. So just to be safe, we'll call her 104000 to make sure we don't steal somebody else's student ID. But we probably aren't going to use this. It's just a unique identifier for her. So, and for all of our patients, or students. So we will assign her the 104 and sort of move on. Okay. So now we've taken care of that particular problem. Let's look again. Let's go back to our structure and understand. So everything that we did is, we cleaned that up for her and for all of the observations that were off for her, but that didn't actually do anything to change the fact that that age was still recorded as character. We didn't do anything to coerce that back to numeric. So let's go ahead and take a look again at students' age. Now we see the only problems we have are a couple of missing values. That's fine, but now they all look like numbers. So we should be able to run the as.numeric function here, and that will coerce the student's age to numeric. It's going to give us an error message that there have been NAs caused by coercion, but we already have the NAs, so we know that's going to happen. That's fine. We don't have to worry about that message too much. So now we can, since age is numeric, we can create our histogram. And now here, this does look like I expected it to look. This is synthetic data, so I didn't really work too hard to balance out those ages, but we see most of our participants are between 18 and 19, another peak between 23 and 24. That could be something like undergraduates and graduates. Our sample does look like a little biased of a college and what the ages should be, but at least these are still pretty reasonable. So now let's move on to another variable here. Let's take a look at the choose your gender variable. And we expect that one to be a categorical type variable with only a few responses. So a table would be an appropriate type of thing to take a look at that. And sure enough, we see female and male. There's a lot more females in this data set than males, but what I'm really looking at right now is that we only have two variables here, or two possible observations here, female or male. There's no typos, there's no any other issues here. So as far as data handling is concerned, this looks like a fine variable. When it came time to actually do your study, you might want to discuss the extreme female bias in this data set. But right now we just have hats on of data handlers. So we're just looking for cleanliness and that everything makes sense. Peeking on also to current year of study. So this one does look like it's got some issue going on here. Some of the year one statements are lowercase and some are uppercase. So we could clean that. I will clean that later on in this script. Right now, it would be nice to move on a little bit. Okay, so let's talk about some of the typos and missingness, things that happen really often that will cause you problems. I guess I did, I apologize. I guess I do clean this variable right here. Okay, so for the student's current year of study, we want to clean up the lower cases into upper cases. We can do that again using that same bracket notation. So if we interpret this sort of from left to right, I'm looking at the variable students, your current year of study. And then within that, I'm applying a logical test again. For all students, your current year of study equals exactly by a logical test, lowercase year one. Then reassign those values to an uppercase year one. So let's run that. We won't see anything happen. I didn't ask for any output, but now I will ask for output because I'll ask for a new table, students' current year of study. And now we see the lowercase year one has been removed. We have nothing but uppercase year ones. So this is an opportunity to really make sure that your code did what you wanted it to do. Because in the previous table up here, we saw year one lowercase of 41 and year one uppercase of 2. So we expect when we merge those two possible values that it should be 43. And that is what we see. So I've also provided you here with an opportunity to do a little code test to understand what my code was doing, to actually see those logical test results and how they're being applied within the coding. Okay, but we saw that our code worked, so we can go ahead and just kind of cut and paste and change the year ones here on the far right to 2s and 3s and 4s, and we can run that and that will clean up our variable. Let's go ahead and check that because so far we didn't ask for any output. And now we see that we have 4 years, year 1, year 2, year 3, year 4 with consistent labeling. And so another little tip here is just test your code as often as you need to be sure it's doing what you want it to do. You can always delete these small little tests out of your notebook later to make the code look more streamlined, but it's really important that your code is doing what you intend it to do. And that way that you can trust the data that you are creating and generating. So now we have created several categorical or a categorical variable with 4 categories, and it would be appropriate to do a bar plot to visualize that. And that's what that looks like here. We have mostly year 1 students, followed by years 2 and 3, and very few year 4 students. But, you know, that kind of goes along probably with the bias in this data set in the first place, that there were so many more females than males. We definitely didn't take like a very good sampling of the college. Or maybe we did, I don't know, maybe we have a lot of attrition in this college, and that's unfortunate. Okay, so another important thing that we really want to understand is missingness. We'd like to know how much missingness do we have. So here I'm going to call a function is.na on the entire data set. So I'm saying which one of these are an NA, which one is a missing value. And that will return another sort of logical test of trues and falses. And when I go to sum the output of is.na students, I go to sum it and add it together. That's looking for numbers, so it will coerce the trues and the falses into 1s and 0s. So basically what this is doing is saying that if you are an NA, return true, which equals 1, sum of that is the sum of all the missings. So when I run that, I see 115. Okay, 115, but I don't remember how big this data set was, and you probably don't either. So what does 115 mean? Well, we're looking at the total number of cells within the data set, which will be the total number of rows times the number of columns. That would give me the entire number of cells in this data set, and it's 1,575. So that means the percent missingness we're looking at is the sum of those who are missing divided by the total number of rows times 100%. And when we run that, we see it is about 7.3. And 7.3% missingness isn't that bad, but a lot of AI algorithms can't handle any missingness at all. So we're going to spend some time trying to clean up that missingness. The first thing that I like to do is understand if there are patterns of missingness. Is there missingness associated with certain variables that were collected, or is it missingness associated with certain observations that were observed? What's going on? So a great function for that is apply, and apply is just going to apply a function of your choosing across your data set, which is students, either the rows, which we would put in a 1 here, or the columns, in which case we would put in a 2. And I try to remember that like the bracket notation where the rows come first and the columns come second. And then the function of your choosing. And you can just put in here a very easy function like sum, and you would type that in as sum with the open brackets. But in this case, I want something a little bit more complicated. I want the sum of the NA values. So I'm going to say function in taking in an input of X, just an input like in algebra, an X, and then a space, and then my little simple function, and it needs to also include that input of X. I wrote those instructions that I just said a little further down. Okay, so running that out, what I see is in the choose your gender, I have 1 missing value. In the age, I have 2. In the course of study, I have 3. These are all very small numbers, except do you take any medication for mental illness? And there is 96 missing there. So that is probably, to do with that variable. Do you take any medication for mental illness is maybe not the most couth way to ask that question. And so maybe that's the point where a lot of people just kind of waved goodbye to this data collector and walked away. So let's see, given 96, how much of that column is missing? So 96 is what we observed, divided by the total number of students. So we got 96 NAs out of how many of our observations times 100 to get a percent. 91% of that data is missing. So with less than 10% of respondents answering this question, there's probably not a lot of information that our model would be able to gather out of this. It's not that less than 10% have are taking medication and 90% are not. It's that less than 10% even answered the question. So whatever pattern might be in there, it's going to be really hard to, pull that out and apply it to the rest of the group. So I'm going to go ahead and remove that piece of data. And that might be something in a repeatable survey. This is an opportunity for you. Maybe this is a pilot survey, an opportunity for you to see, oh, I didn't word that question correctly. If I reworded that question, maybe I would get some better responses out of that. But in the meantime, I'm just going to remove it. So again, I'm applying a logic test with the which. I'm saying which names, the column names here of the variable is, do you take any medication for mental illness? And then students, then we're going to remove that column, and it is the 15th column. So let's run this here. Sorry, I should have separated it into 2. So the which name students, which column name is, do you take any medication for mental illness? And it's number 15. So I'd like to remove the 15th column. And here I'm using the minus sign to say that is the one I would like to remove. Removing by name is actually kind of tricky, so that's why I'm removing by number. But I can say that here in a little pro tip box, that if you don't want to delete it by number, because that is technically something called hard coding, then you can do this by using a variable name. So the problem with this hard coding example is if I did something and changed my notebook and suddenly do you take any medications for mental illness is not the 15th column, R doesn't care. R will delete the 15th column, whatever it was. So instead, you can use that which notation and assign it a name, and then you can run the next column by name. And that way, instead of looking at the 15 and hard coding it right here, I'm saving it as something called meds, and that could be 2 or 8 or 12. And then this line will run and remove 2 or 8 or 12 or whatever it is. Okay, so going back to our missingness notation, now I have deleted that column. So now let's redo my same missingness calculation and see how much it improved. So now we're down to 1.3% missingness, when before we were at 7.3% of missingness. So just removing that column of data did a lot to improve our missingness. So now let's take a look at the row missingness, because the other ones all only had missingness of 1 or 2 or 3. And so that's probably not systematic to the columns. It sounds like that might be systematic to the rows. So I'm going to do that same apply function again, where I'm going to apply a function of my choosing to students across the rows this time, which is the ones. The function I'm going to create is going to have a variable expected input of X. And my function here is going to be, let's look inside all these parentheses, which X's are missing, divided by the total column number of students. So I'm looking at the rows this time, and the missingness across each of the observations. So across the total number of like questions I ask that person, how many are missing, times 100 to get a percent. And then I'd also like to round that percent so that it comes out looking a little easier to understand. So the round function here, I am taking an input of 2, saying I want to keep 2 points after the decimal. So when I run that, I see that for most all of my observations, they're 0. They have no missingness whatsoever. As I scan along here, when I get to observations 103, 104, 105, I see very high amounts of missingness. So what I see here also is there's really a lot of numbers. And so there's kind of more than I maybe feel comfortable scanning with my eye and then saying, let's specifically look at 103, 104, 105. A lot of them are 0. So let's go ahead and do a quick function here to understand the missingness a little better and omit those zeros because those are complete data cases and we don't want to worry about that. So I'm taking the same function that I just did right here, the apply function, but this time I'm giving it a name so I can keep this output and I can manipulate and talk about this output. So I run that. And then now we can visualize the row missingness here. And so like we see, there's a whole bunch of zeros and then a couple of guys that are much, much bigger. And this is the kind of histogram that I mentioned in sort of the lecture side on the slides that doesn't look right. This one is definitely telling you somebody is an outlier. So here, let's take a look at just those rows that have values greater than 0. So I'm using that bracket notation again. I'm applying a logical test for those row missing values whose row missing value is greater than 0. That's what I would like to see. And there I see observations 44, 103, 104, and 105. And I'm really glad I did this because I didn't see 104 and all that output from before. That one didn't just pass right over my eye. So now we know we actually have 4 that we need to take a look at. Let's take a look at student 105 because 64% missingness, that's probably more than we can handle by any sort of imputation or anything. That sounds like a lot of missingness to me. And taking a look at that guy there running that cell. In fact, it looks like all we know about this person is that they are 19 years old and their data set was collected on the 19th of January, 2021. So maybe this is a point of data collection where somebody flagged down a person. Hey, can I ask you a question? Sure. How old are you? 19. What's your gender? I don't want to answer that. And they left. So if we only know that the person is 19 years old, there's really nothing I can do with that. So I'm going to go ahead and omit that person. Now we omit that person by saying the student minus the 105th row, because we're talking about the row here, and I'm going to reassign that to the variable students. Another thing that I could do is give this a new variable name. I tend to do that a lot. So I could call it students 2. And then from here on out, you would want to change every single piece that I have written all the way down using students 2 instead. For this particular notebook, I didn't do that and I don't want to cause errors, but I like to do that because it doesn't really hurt me to keep this extra object in my notebook environment of the original students. But now since I've started to make edits, I give it a new name. So that's something I do a lot just so I don't overwrite my code. But so we run this and that has removed student 105. Now let's take a look at student number 104. And 104 is a 20-year-old male in his 2nd year of college. He's unmarried and he didn't seek any treatment with a specialist, and I have the date. So, perhaps that we could, impute some kind of, some of this information based on other males in the sample. But we don't really know a lot about him. We only know that he's 20.
Courtney?
Yes.
We're now like 40 minutes over. Can we not do each individual and sort of wrap up how to do this?
Not really. I guess feel free to click through the code on your own, but the rest of the cells won't work if we don't run these.
Okay.
Cells. But I mean, everything I'm saying is right there in the markup before and after each of the cells. So you're free to go through the rest of this workbook on your own.
And the workbooks are there for all of you to use. They will never disappear on the ScHARe platform, so you can do that. I really appreciate that many of you stayed on for longer than we had originally planned. And I especially want to give a thanks to the 3 guest presenters that we had today. Even though what Courtney was kind of doing might have been a little bit over some of your heads, and it was going a little bit fast because we had lots of different things to do. So I hope that I see in the chat that some of this was a little bit over certain people's learning levels. I think the take-home message with this is that these notebooks, as you saw her do, she never had to type anything. All she had to do was click on the cut and paste. And this is what we've tried to do with the ScHARe platform to make it easy for everyone to use. These notebooks are just cut and paste. So even though we didn't get through the whole presentation, you can still work on it on your own and just cut and paste. And many of the notebooks that are now on the ScHARe platform have many different options like she was doing. And there were lots of questions in the chat earlier about missing data, and she went over all of that on how to fix it or what to do with it or how to find it even in the presentation and the demo. So please use the ScHARe platform. Please use the notebooks to help you do, to learn. Because once you learn how to do this, then you'll end up writing your own code and doing that kind of stuff. But the process of learning, there are tools on the platform to do such. Elizabeth posted in the chat where these will be posted on the NIMHD webpage. It takes a couple of weeks to get them actually posted because everything has to be 508 compliant, but they will be there and they remain there. So you can go use them anytime that you want. So I'll ask my co-presenters and Luca, if you have any closing last words that you want to add.
Oh, thank you. I would just point everyone to the list of tutorials and resources that we have on our NIMHD website. They could be helpful.
And I especially want to acknowledge and thank Rebecca Rodriguez from NIDDK for helping with this. And I encourage all of you to at least look into the challenge and apply if you can. So thank you, and we'll see you in a few months. Oh, I guess we won't see you for the holidays, so happy holidays.